Hi, my name is Eli Lyons. I'm the Chief Data Officer at Molcure, and today I'm going to explain about our company Molcure. Antibody discovery is very important for therapeutic drug development as well as research purposes. And one method of antibody discovery is through phage display library screening. So, in the conventional framework for antibody discovery using phage display library, there are many steps which are time-consuming and a lot of labor, and there's also a lot of bias in the experimental framework, particularly at the antibody selection and amplification. This is cycled through many times, and the bias is compounded, so a lot of good antibody candidates never get discovered. There are multiple methods for antibody discovery, but what they all have in common is they require a lot of time and manpower, and there's a lot of bias in the results. So for example, the hybridoma method requires a lot of time, and the results, there's low probability of success. And immunization animals with an antigen, this also takes a long period of time, and still the antibodies will later need to be humanized, which takes more time and also results in lower success rate. Our solution is a rapid antibody engineering system we call AbTracer. AbTracer is a patent-pending framework which reduces the number of steps from the conventional framework. It uses high-throughput next-generation sequencing and antibody big data analysis to find better candidates than the conventional method. So step one of the AbTracer system is the creation of a phage display library. In the phage display library, the antibody coding DNA sequence of the phages is engineered so that a whole library is created of phages with different displayed antibodies on their head. So you want a large variation in this library. In step two, a phage library can be screened against one target antigen and successful phages can be purified. Or a phage library can be made to screen against mixed and normal targeted cells and do cell sorting to isolate the target cells and antigens specific for that population. In step three, we perform antibody DNA sequencing. Here we sequence the phages before screening and after just one round of screening, so the experimental bias is reduced, and we use high-throughput next-generation sequencing to identify key antibody coding sequences. In step four, we do an antibody big data analysis, which is really important for discovering good antibody sequences. So we take the antibody sequences, we perform clustering by similarity and also ranking, and we do de novo binding factor analysis, which is basically simulating binding data. We also do machine learning, which is based on previous screens from our database. And all of this together allows us to identify the best drug candidate antibody sequences. AbTracer has several advantages over the conventional framework. We can detect antibodies with higher diversity, and it's faster than the conventional framework. And by doing only one round of panning, we reduce the bias inherent in the system. It's also flexible. It can be used with immune and non-immune libraries. Currently, we offer a service to our clients where they provide the before and after panning antibody library, and we can do sequencing and data analysis and find the best antibody drug candidates. In the near future, we hope to create our own libraries and perform the screening by ourselves. In conclusion, AbTracer can add a lot of value for our clients. We can provide our service and find candidates much faster. We can find novel antibody candidates that wouldn't be found otherwise with high affinity, high specificity. It's flexible. We can use immune and non-immune libraries. For the same price, we can find 10 to 20 types of lead antibody candidates, and whereas the conventional method may only find 1 to 2. The antibody drug market is already a $45 billion market, and it's still growing a lot. And so we want to have a high impact in this market. This is our 5-year plan, and in our first 2 years, we'll initially only offer our antibody screening service. But in the 3rd year, we'd like to enter into collaborative work with another pharmaceutical company to fully develop an antibody drug. So the majority of our clients are based in the US. So we have a marketing strategy for both pharma and academia. For pharma, we have a direct sales marketing strategy where we contact potential clients directly. And for academia, we plan to partner with other distributors to help us with sales to various locations. AbTracer requires development in bioinformatics, biology, and robotics for automation. Our team is extremely multidisciplinary, and we have expertise in biology, IT, robotics, and business. To further develop our business, we plan a horizontal expansion into screening other molecule types such as peptide libraries and aptamer libraries. Thanks for listening to our presentation, and we look forward to getting feedback from you.